Welcome everyone to join our Nature Nanotechnology 15 years anniversary webinar. It's our honor to have Professor Hagan Bayley from the University of Oxford and Yamuna Krishnan from University of Chicago to join us. Now we will start with Hagan. So Hagan Bayley is a professor of chemical biology in the University of Oxford. He was trained as a chemist, but interested in working from a more multidisciplinary perspective. He did lots of studies and engineering on the transmembrane pore-forming proteins, as well as chemical signal transduction and biomolecular materials. He's also very good at commercializing basic science. His research has produced 2 spin-off companies, including the very well-known Oxford Nanopore. Today, Hagan will share with us his research on Nanopore, from gene sequencing to organ repair. Hagan, please go ahead.
Okay, thank you. It's a real pleasure to be celebrating 50 years of Nature Nanotechnology. I went back and had a look. We've actually published 9 papers in the journal, which is very nice. I won't tell you how many we've submitted. Sometimes the journal can give you a bit of a hard time, but it's very nice to have been part of the 15-year history of the journal. So I'm going to give you really an overview of what we've been doing with nanopores. I'm not going to go into any really extensive detail, just try and show you the big picture. So what do we mean by a nanopore? We're going to be talking about protein nanopores. Well, if you go and get your COVID injection, you'll probably be injected not with a 21 gauge needle, as I'm showing here, maybe 23 gauge, maybe something a little narrower, and that's about 500 microns in diameter. A blood capillary human hair by comparison, actually they vary quite a lot, would be about 80 microns. And then we can get down to smaller and smaller diameters. A blood capillary 5 microns, nuclear pore 30 nanometers. But the kind of pores that we're dealing with have diameters of 1 or 2 nanometers. They're really are diameters of the molecules that we'll be interested in, that I'll be telling you about. So they have about 1/50,000th of the diameter of a human hair. And I hope to convince you that they have interesting applications both in basic science and in biotechnology. So where do these pores come from? Well, as many of you know, cells are bounded by membranes and divided into compartments by membranes, and those membranes contain many interesting membrane proteins, proteins involved in signaling, enzymes, transport, ion channels, and so on. But our interest is in pores, and there are a wide variety of protein pores, and they can have a larger diameter. I'm showing the pore Streptolysin O from Streptococcus here, and that's a 100 nanometer scale bar. This particular pore would contain maybe 50 subunits and is used by bacteria to attack humans. But humans also have pore-forming proteins, perforin and C9, for example, which we can use to attack invading organisms or infected cells. So the pore that we've looked at the most, although we've looked at several different pores in the lab, is called alpha hemolysin. This pore has a diameter of a couple of nanometers, and it's secreted by Staphylococcus aureus, again as a pore-forming toxin, and each subunit has about has exactly 293 amino acids, and it's made as a water-soluble monomer, and it assembles in membranes to form pores with 7 subunits. And the structure of the pores was solved almost 25 years ago now by Eric Guo's group in collaboration with our lab. And you can see that on the left, we'll come back to that in a moment. And on the bottom is AFM view of the top of the protein, where you can pick out the 7 subunits. So one thing I'd like to emphasize is really that our work in the biotechnology area really began as a basic science problem. And I'll emphasize the fact that it's really basic science that underlies all the applications in biotechnology. So the question we tried to answer was, how can a water-soluble protein assemble into a transmembrane pore? And we were able to work out this pathway for alpha hemolysin, which is actually generally applicable to other pore-forming proteins. The monomer binds to the membrane, assembles on the membrane surface, the membrane is broken open, and then finally the pore inserts into the membrane, in this case to form a beta barrel through the lipid bilayer. So really working on this problem provided the knowledge and ideas that were needed to apply nanopores in biotechnology. So I would contend that advances in applied science come through discoveries in basic science, and second, that the science that you need to apply things like protein pores in biotechnology is really highly but multidisciplinary. And today I'll tell you about two aspects of this that were initiated in our research group in Oxford. First, the ultra-rapid DNA sequencing that was developed finally at Oxford Nanopore, and then with some newer work on the 3D printing of synthetic tissues and also living tissues. So what you need to know to study protein pores, but again, I would emphasize this is highly multidisciplinary, and the kind of techniques that we use involve chemistry and biochemistry, as well as cell biology, a bit of genetics, physics, mathematics at a fairly low level, and device engineering. And you'll see how that fits in as I go along. So one of the really important measurements that we need to understand is single channel electrical recording. And in general, we do this in planar lipid bilayers. So we will make a lipid bilayer, insert a single copy of a pore, such as the Hemolysin pore, into that bilayer, and then apply a potential, and a current will then flow through the pore. And that current is carried by ions, not by electrons, obviously, so it might be carried by sodium and chloride ions, for example, or potassium and chloride ions. And these currents are quite small. They're in the tens of picoamp range under our normal conditions, but by using nice quiet patch clamp amplifiers, it's possible to measure changes in these currents down to a few percent. And so these would be a 10 billionth of a current that's in a old-fashioned incandescent light bulb. So here's our protein pore again, typical of the pores that might be used in sensing and sequencing using nanopores. And the important thing is that the internal dimensions of the pore. So if we enter at the top on what we call the cis side, this entrance would be about 3 nanometers in diameter. There is a kind of vestibule in the middle, and then the part that goes through the membrane is roughly 2 nanometers in dimensions. And the important thing about this is that it will fit in small molecules, as you'll see in a moment, and also polymers can pass through the pore. So for example, single-stranded DNA will pass through the Hemolysin pore, but double-stranded DNA gets stuck. So in other words, the dimensions of the pore are similar to the dimensions of molecules that we're interested in looking at. And this led many years back to the idea of stochastic sensing with protein pores, which is a single molecule detection technique, and initially based on non-covalent chemistry. So the idea is shown here. I'm showing a single pore in 2 states. It will have a binding site inside it, and when a molecule binds inside the pore, it acts as a partial blocker and changes the current, the ionic current that's flowing through the pore. So we might see a trace like this where something binds, comes off, binds, comes off, binds, comes off. And this is a stochastic process. And so not all the binding events are of equal duration. They're exponentially distributed. But the frequency at which these events occur and will tell you the concentration of the analyte that you're looking at, and this can be used with a wide variety of analytes, and the amplitude of the current block and say the noise during the current block and so on will tell you something about the identity of the analyte. So I'll show you one example here. I won't go into this in detail because this is pretty old work, but here's our Hemolysin pore. It's been engineered in this case so that a cyclodextrin molecule will bind inside it. And beta cyclodextrin has the same C7 symmetry as the pore. It has 7 glucose subunits organized in a ring. Sometimes if I'm talking to physicists, I will tell them this is a three-dimensional structure of cyclodextrin, but I know for the readers of Nature Nano are a bit more sophisticated. But anyway, this is a bit like a molecular grommet that fits inside the pore, and this will bind organic molecules. And in this particular case, we're looking at ibuprofen, Advil or Motrin, and this molecule comes in two optical isomers, the R and the S form. And it's the S form that's actually an active pharmaceutical, although when you buy ibuprofen, you get a racemic mixture of the two. And when we add this to one of the chambers of the device that I showed you earlier, you'll see, in fact, 2 types of binding events. So there's a binding event due to the R form, binding events due to the S form, and you can distinguish those because the cyclodextrin is chiral. So these are non-covalent diastereomeric complexes, so they're physically different. And you can just distinguish the mirror images of this molecule. So it's a very sensitive detection technique. Further, this trace here is about 50 milliseconds across. So this can really work on a rapid time scale, hundreds of microseconds upwards. So having got a lot of results like that, we could also look at metal ions, peptides, you name it, anything that dissolved in aqueous solution, we can detect by this technique. We thought, well, antibodies and enzymes have been very useful in biotechnology. Maybe protein pores would have some useful applications. And based on that, a long time ago now, in 2005, we formed Oxford Nanopore. And I always like this description of Bayley speaks in the laconic baritone of a late night radio DJ. If he has an entrepreneurial streak, it's not obvious, but I think we had the last laugh just now in September when finally, after many, many years, Oxford Nanopore was IPO'd and is now has almost 1,000 employees and is valued at about £4 billion. And this company was formed in Oxford. The CEO is Gordon Sanghera, who worked with Alan Hill on the glucose sensor, and Spike Wilcox was both an undergraduate and a PhD student at Oxford, and was there at when the company was first formed. So when we first formed the company, the idea was to detect small molecules, so-called biomedical markers, and that was our game plan or business plan to start with. But there'd always been this idea around that you might be able to sequence DNA using nanopores. This was really quite a speculative idea, and up to that time, really no substantial work had been done in the area. And there are 2 ways you might do this. The one on the right, I think, is still a viable possibility, is that you could use an exonuclease to snip off one base at a time, and then just as we can detect ibuprofen, you would detect these nucleoside monophosphates as they're clipped off. And then the other method, which is the method that finally worked out, is that you could run a single strand of DNA through the pore, and it might go past a constriction, and then you could read off each base at a time, that each base might have a characteristic current level. Of course, it turns out to be a lot more complicated and more difficult than that. But the point is that we're sequencing unmodified DNA, no fluorescent reagents or anything used here. It's really the raw DNA that's being sequenced. So a lot of the initial work, which I'm not going to go over, was done in academia. So we had to show that the DNA could bind and be captured by the pore and improve the efficiency of that process. We had to show that the bases could be identified just as small molecules can be identified. And we also had to devise ways to translocate the DNA through the pore, because one big problem with this technique was that once you get DNA to move through the pore, once you apply a high enough potential to get it to move through electrophoretically, it moves through incredibly quickly at about 1 to 10 microseconds per base, which is too fast to do the base identification. And then there were other goals. We, as you'll see later, figured out pretty early on that a single pore wouldn't be good for sequencing, say, a human genome, and arrays of pores had to be assembled to do parallel nanopore sequencing. But that part was initiated and completed at Oxford Nanopore. So a lot of this initial work was done in academia, and then finally the company really took this to completion. And skipping over many years of work, it was really 7 years in the company, so we needed long-term investors to be able to get this done. The MinION sequencer was developed. There's an early version of it here, stuck together with a bit of tape. But anyway, the version that we have nowadays is very cheap. It was initially just given away for free, and that you paid for the reagents. It's obviously highly portable, which is really a revolution in nucleic acid sequencing. You can do ultra long reads. The record now is about 4 megabases, which is really quite phenomenal. And as you'll see in a moment, this device has hundreds of pores sequencing in parallel. And recently, the accuracy has been hugely improved, and we'll come to that in a moment too. So the first thing that Oxford Nanopore really improved was the translocation of DNA through the pore. And nowadays they use a helicase to do this. So a helicase will take a double-stranded piece of DNA and in a energy-dependent manner, ATP-dependent manner, will split it into 2 strands, and during that process, it can pump one of these strands through the Hemolysin pore. So by using the enzymatic technology, this really slows down the DNA. It's a ratcheting motion. It moves one base at a time. So you move, have a look, move, have a look, move, have a look, and that's important. Formally, also, if you apply a potential, it stretches the DNA. That's important too, so it doesn't coil up inside the pore. And also very important is the fact that it uses double-stranded DNA. Single-stranded DNA is it forms very viscous, messy solutions. So you'd obviously like to work with double-stranded DNA. So this was a big development, really, in DNA sequencing. And the next thing was the base calling. We'd already shown in academic labs with immobilized DNA that we could distinguish the 4 bases, C, A, T, and G, and also modified bases. But Nanopore developed sophisticated methods to deconvolute the signal for DNA as it moves through the pore. And the important thing is, if you have a particular base C, it won't give, all the C's won't give the same current level. The current level that they give depends on the neighboring bases. So it wasn't as simple as I showed earlier to deconvolute the signal. And in the early days, basically they used a hidden Markov molecule model, to deconvolute this signal, which you can see the individual steps as the DNA is moved here. And the idea was that if you move the DNA by one base, there were only really 4 different current levels it could move to, because of the 4 different bases, and then so you could identify that current level. And then, of course, oversimplifying, if you moved it by one base again, there were only 4 different possibilities, and that allowed you to deconvolute the signal. Now, in the last couple of years especially, by using much more sophisticated signal processing techniques, the accuracy of the base calling has been hugely improved. So even about 3 years ago, it was still around 92%, but nowadays it's up to 98%. And this development, most of this development really just happened over 2 years. It's huge improvement in so-called base calling. Now, I've emphasized that there's also a need for a multiplex system, and I think the $1,000 genome, which was a goal when we first opened the company, was really brought to fruition by Illumina and other ways of sequencing fairly early on. But the goal then became to do faster and faster sequencing. And the human genome is about 6 × 10⁹ bases deployed genome, and at 10 milliseconds per base. Actually, the device is now sequencing at about 2 milliseconds per base. It would take a single pore about 2 years to sequence the human genome, and 10,000 pores in parallel take about a day. And as you'll see in a moment, that's where we are approximately at the moment. If you could make a device with a million pores working in parallel with a 10x coverage of the sequence, it would just take you 10 minutes to do it. And I think really within the next decade, we'll certainly see that. And of course, a bacterial genome can be sequenced incredibly quickly, which in terms of identifying bacteria in a biological warfare situation or in an infected person will be incredibly valuable. So Oxford Nanopore developed a chip that was based on an application-specific integrated circuit. So the original chip contains about 2,000 little wells. About 500 of those are active at any particular time. And by using various tricks, you can get close to 1 nanopore in each of these apertures. And that allows you to sequence from 500 pores, so you can get about a gigabase per hour from the chip. And the development of that chip, I think, was a real tour de force by the company. This was not done in an academic lab, and really involved micropatterning of the surface and using a block copolymer rather than a lipid bilayer to form a much more stable film that the pore could go into. And here is a animation probably made at great expense by Oxford Nanopore to show how the system works. So you have all these wells, hundreds of wells, each one containing a single Hemolysin pore, and your helicase will come along holding a piece of double-stranded DNA, and then the enzyme is released and goes into action, and a single strand of DNA goes through the pore, and you get an electrical signal which can be converted to a DNA sequence. So basically, at the end of all this time, what has the company come up with? This portable device, parallel sequencer, the MinION. A lot of the development has been really open development. So sequencing data, software improvements, sample prep procedures, and so on are shared on the internet, and there's a really quite large community of users that share this information. And this has now appeared in many scientific publications, including Nature Nanotechnology. And I always like to show the first public release of sequencing data from the MinION that was done by Nick Lohmann's group at the University of Birmingham, who's been a big player in this area. And this is the original trace that he obtained, and here's the actual sequence. This was I think around 8,000 base sequence that was I mean really long at the time. And I have to apologize to the people at Nature and Nano, because the venue of choice for publishing this information was actually Twitter. This is where this first came out. After that, things moved pretty quickly. So Nick's group and other people used the MinION to sequence viruses, and of course it's a portable device, so this could be done in the field. In 2016, during the Ebola crisis, Ebola virus was sequenced. Of course, there's no such thing as Ebola virus. There are hundreds and hundreds of different Ebola viruses that can be linked in a phylogenetic tree, some of which are worse for you than others. But obviously these small genomes could be sequenced very quickly. And the next year, during the Zika virus epidemic in Brazil and elsewhere, the MinION was also used. And then soon after that, the device was used, or many of the devices together, were used to sequence larger genomes, so human genome in 2018, and also the ability to sequence highly repetitive sequences was demonstrated in that time, and also RNA sequencing, which of course is hugely important nowadays, was also demonstrated in 2018. So what's happening nowadays? Well, nowadays, as well as the MinION and even smaller devices that are under development that could just clip onto your phone, there are large throughput devices now being sold by Oxford Nanopore. So this Prometheon device contains 48 flow cells that would be similar to the ones in the MinION. They contain more nanopores, so they can have up to 3,000 pores sequencing simultaneously. So this device would have 300 times the capacity of the MinION, and in a couple of hours can sequence about 15 terabases. And this allows you to get a sub $1,000 human genome pretty readily. But whoops, let's go. Sorry. So even at quarter capacity, which is what I could get the data for, this device can sequence 2,000 COVID genomes in a day, and so far about 400,000 COVID genomes have been sequenced using Nanopore sequencing. So I'll just sort of go over a few recent developments, which I find pretty interesting. The Hemolysin pore is no longer used by Oxford Nanopore. The pore they use now is called CSGG, and this just shows the really curious link, I think, between basic science and applied, biotechnology. And the CSGG pore is actually a 9-mer. 262 residue 9-mer, so it's a 36-stranded beta barrel, has very interesting structure, and it's found in bacterial outer membranes and can secrete amyloid-like fibers onto the surface of the cell to produce a biofilm. So this was really, at the time it was discovered, was really nothing to do with nanopore sequencing, but oxynanopore made over 3,000 mutants of this pore, and it eventually became the pore of choice, having this narrow constriction for sequencing DNA and RNA. Another development that's about to come to fruition is rereading single molecules. So you can capture a single-stranded DNA in the pore, it would come from the helicase, and you can sequence it, and now it's possible to, by using sort of feedback technologies, to pull that DNA back and sequence it again, and you can sequence it again and again and again and again, and that will allow you to obtain incredible accuracy where that is needed. And then when you don't need that piece of DNA anymore, you can just eject it from the pore. Okay, so what does the future hold? Well, right now there's a lot of interest in using nanopores to look at proteins. As you know, there are about 20,000 human genes. They form a huge number of transcripts, and these transcripts can be made into millions of so-called proteoforms, which cannot be predicted from the sequence of the genome. And these proteoforms come from post-translational modifications, in alternative splicing of the transcripts, and so on. So one goal that where there's a lot of action in the field at the moment is to be able to count the number of these different species, maybe in a single cell, and to be able to map the post-translational modifications and splicing in individual molecules rather than get population distributions. The last thing I'd like to show you about sort of nanopores working with single molecules is that we can also do chemistry, and I'll just go over that really very briefly. So we can look at bond making and bond breaking reactions, not what I've shown you before, obviously non-covalent interactions. And I won't go over the actual chemistry here, but here we have an arsenic compound binding to a sulfhydryl group that's inside the nanopore, and that will change the current that goes through the pore. And then there are various different ligands that can come on and off the arsenic. And it turns out that that gives you a reaction network with 38 possible reactions. And by analyzing the transitions between these different states, you can tell which reactions are allowed, the green ones, which aren't, the red ones, and get the rate constants for all of the reactions in this complicated network. And the kind of data that you will get that allows that kind of analysis is shown here. So it's really quite beautiful, almost like some sort of musical score, and you can read off the kinetics, or you can compute the kinetics using this. And another thing that's Yujo King in the lab has done recently is develop molecular walkers that can step, well, hop between sulfhydryl residues on beta strands in the pore. And that enables you to move a piece of DNA through the pore, turn it around, and then move it upwards, turn it around, and then move it downwards again. And the chemistry involved is very, very simple. So it's just a disulfide interchange. So the piece of DNA is sitting on this cysteine residue here. It's going to move to this one and then to this one, as you'll see in this animation, just by breaking of this bond and formation of this bond. So there you can see it go. Yeah, and what we'd like to do ultimately is get rid of the enzyme in DNA sequencing and use a chemical method for stepping DNA through the pore. And that's one area that we're working on at the moment. Okay, so let me finish with how protein pores can be used to make synthetic tissues. And this was really derived again from another piece of basic science. We wanted to miniaturize our bilayer recordings. Those chambers that I showed you earlier can be ranged from 1 mil down to maybe 25 microliters in volume, but it's possible to do this with droplets that are connected through lipid bilayers. These droplets exist in oil. You form a lipid bilayer between them. These are two silver-silver chloride electrodes. So you can get down to nanoliter volumes and measure currents through pores. So these could be very rare pores, or you could be using very expensive reagents. So we figured that by using multiple pores, we could bring hundreds of these together and make what we call a synthetic tissue. So you might ask, what is a tissue? Well, really a tissue is a collection of cells that can interact with each other and interact with the environment. And of course, these materials have emergent properties that are greater than the sum of the individual compartments. And what we're trying to do is make synthetic tissues, but also print cells, and then bring these together to make hybrid materials. And our long-term goals of this would be just like the sequencing, to greatly reduce the cost of materials that can be used in medical applications. So you might use these to screen therapeutics, to deliver therapeutics, even make them before delivery. But in the real long term, we could use these materials to repair organs, or possibly even fabricate organs at relatively low cost. And I actually got the idea for printing tissues from reading The Economist magazine and seeing that way back 10 years ago now, people had actually printed violins or chainmail gloves. So I thought, well, if you can do that, why can't we print biological materials? And so what we've ended up doing is printing aqueous droplets. Those droplets are separated from each other by individual lipid bilayers. As you'll see in a moment, you can put membrane proteins into these bilayers, and that allows the droplets to communicate with each other. You can use the Hemolysin pore or other pores or channels or transporters to do this. So the general idea is that you have an oil, there's an amphipathic molecule in the oil. That will, if you put a water droplet into the oil, the amphipathic molecule will form a monolayer around the droplet. Then if you bring two of these droplets together, you'll get a lipid bilayer. That's why we wanted to use them for bilayer recording. And we're also able to use this at a Nature Nano paper, my favorite one perhaps, to make small devices. So we could take droplets, put engineered pores in them that only conduct current in one direction, and make devices like this one, which is a full wave rectifier. So it will actually rectify an electrical signal using protein pores that have been put in the bilayers between these droplets. So if we wanted to extend this, we obviously had to make 3D structures that were much larger, and we also wanted to be able to pattern them. So we developed a 3D printer that would enable us to print, in this case, 50 picoliter droplets rather than droplets of 100 nanoliters. And you can see the sort of structure that you will get out, though that's shown better maybe on this slide. So here's one that's printed in 20 layers. It's kind of got an arborizing pathway through it that's shown with this blue dye. And here's another structure that looks different in the X, Y, and Z directions, which gives you some idea about the fidelity of the printing. And if you look very carefully, you can see the individual droplets. Each droplet, if you think about it, I'm not going to go into details now, is actually a dodecahedron. And here's a beautiful picture that was made in the lab by polymerizing one of these structures and then fragmenting it and looking at it by optical microscopy, and you can see the different facets on each of the compartments. So one thing that we're interested in, amongst other things, is using these materials in medical applications. We'd like to have input signals to be able to process these signals and have different outputs. So we'd like to be able to transmit electrical signals, get these to change shape and so on, and make and release drugs. And here's one example here where we can transmit an electrical signal through one of these tissues by putting Hemolysin pore only in the green droplets. And if we bring up two electrodes and apply a potential, a signal is transmitted directionally and very quickly. And that's obviously a different mechanism to how a neuron works, but it has the same outcome of fast directional signaling. We can also do more sophisticated things like turn on transcription and translation in individual droplets. So here we have a promoter that's got modified bases in it, and these modified bases terminate in a biotin that can be cleaved off using a nitrobenzyl group to release the biotin, and that means that we can block this promoter with a monovalent streptavidin, shine light on, and then the promoter becomes active. This is just one for T7 polymerase, and then it will then make RNA, and that RNA is ultimately can be turned into protein. So here you can see how we can turn on a yellow fluorescent protein in a specific region of our printed structure, and that can actually be used to turn on electrical signaling, because we can turn on the synthesis, say, of the Hemolysin pore. So I'm almost finished now and almost on time. So we've been now recently using the same technology to print living cells, and given the revolution, really revolutions in cell biology at the moment, stem cells, genome edited cells, use of personalized cells, this is a really exciting area. So we can capture living cells in the droplets that we're printing with a 3D printer, and then we can print them in different patterns, and by using the lipid bilayer to hold the droplets together when they're first printed, these patterns can be stabilized, and then we can transfer these structures, you can just about see the individual cells here, to growth medium, and at that point the lipid, which is shown by this red dye here, disappears. It either goes off into the medium and is bound by proteins in the medium, or is actually eaten by the cells. But now you end up with a pattern, a 3D patterned array of cells. And just to show you one example here, here are some human neural stem cells that are printed in 3 dimensions. After a day in culture, they just look like these little individual cells. As time goes on, they begin to put out processes, as neurons do, and by day 14, you can clearly see these long axons that have been put out by these individual cells. And one thing that we're trying to do at the moment is print patterned structures of neural cells that can be used to repair the damaged brain, either damaged by traumatic injury or by neurodegenerative diseases such as Alzheimer's and vascular dementia and so on. Finally, we'd like to bring our synthetic tissues and our living printed tissues together. There are different ways we can do that. One is by, actually, you guessed, engineering pores. So here's a Hemolysin pore where we've made a dimer of heptamers. So there's a membrane spanning region at each end of this dimer. This is a class average that's shown here. And we can use this to link structures such as these liposomes together with pores that are inserting into both bilayers. And that might be one way to bring these synthetic tissues which don't contain DNA, can't divide, and whatever, together with the printed living tissues. So we can make both synthetic and cellular structures at high resolution by 3D printing. We can make hybrid structures, and now we'd like to turn our attention to applying those in fundamental biology and medical and surgical applications. So basically, the story that's gone in parallel with publishing in Nature Nano is that we started some very basic science on membrane proteins that allowed us to develop single molecule sensors, which led to Oxford Nanopore and DNA sequencing. By again doing some basic science and trying to miniaturize recordings, that led to printed droplet networks and another company, Oxybio, that's been used to exploit this technology. So if people often ask me, what do I have to do to start a company? And I think the best advice really is to do some really great basic science. So when you come to commercialize it, it's readily fundable. And I'll end by thanking the people in my group. Here they are, so the aged members at one end and the younger members at the other end. And thank you very much for listening. Thank you.
Thank you, Hagan. Thank you for sharing with us the knowledge of nanopores and synthetic bio. So we don't have much time now, so I'm going to ask some general question on behalf of our audience. So after talking about so many great achievements of nanopores, do you want to share with us about its limitations? So for example, people are concerned about the stability of the protein pores. That's why lots of people are also studying solid state nanopores. So do you have any thoughts about this? Any suggestions for the people working on solid state nanopores?
Well, if it was a very short suggestion, it would be stop. But more seriously, I think solid state pores are you can't manipulate them with the precision that you can manipulate protein pores. So we can, obviously, by using site-directed mutagenesis and directed chemical modification and other techniques, we can engineer protein nanopores very precisely. Furthermore, every one that you make is the same. There are some caveats to that. And also, I think the idea that they're unstable is really incorrect. So the alpha hemolysin pore, for example, we can do recordings from that at close to 100 degrees centigrade when there's steam coming out of the apparatus. So these pores are perfectly stable. In fact, it really for Oxford nanopore, it's the lipid bilayers that were really the weak point, and that's why they were replaced by block copolymers. And I think finally for the solid state pores, they're obviously stable at several thousand degrees, but I would not advise sequencing DNA at those temperatures. So I'm a great believer in protein pores, and they're certainly adequately stable for just about any application you can think of.
Thank you, Hagan. So now I will leave the stage to Chiara Pastori, my colleague, and she will introduce Yamuna Krishnan later. Chiara, are you ready?
Yes. Hi. Hello, everyone. And yeah, I'm Chiara Pastori, despite the name on the screen. Thank you very much, Professor Bayley, for your really great seminar. It was a pleasure to listen to. And before I introduce you to our next speaker, I'd just like to talk a little bit about these webinar series that we have organized for the 15 years of Nature Nanotechnology. Today's event was the 4th webinar in a series of 7 webinars. So there are still 3 to go, and you can register for free to all the remaining webinars, or just to one of them if you want to, by clicking on the link that is provided in this slide, which is also the link that you've used to register to the current event. You'll also receive an email shortly after the end of today's event with the link again. So you just have to click. Once you click, you should have the choice of register for one specific webinar. And so next one is going to be next Wednesday, and it will be focused on energy storage and electrochemistry. So 2 topics that are really important for the future of our society, really, and 2 topics where nanotechnology can really play a big role. So if you're interested, and we hope you are, then just click on the link and register for next week's event. And now it's my great pleasure to introduce you to our next speaker, Professor Yamuna Krishnan. Yamuna is a professor at the University of Chicago since 2014, I believe, and she leads a lab that specializes in designing small DNA probe that she uses to investigate the chemical reactions happening within organelles in a cell. So really to provide a quantitative imaging and mapping of the chemical status of these organelles. And the idea is, of course, to gain a better understanding of cellular biology, but also to use these as diagnostic tools to have fingerprints of the chemical status of the organelles and to eventually maybe use this for designing specific precision medicine strategies. But she will present all of this, I think, in her talk that it's entitled Exploring Living Systems with DNA Nanotechnology. So thank you very much, Yamuna, for being with us today, and I'll leave you the floor. And you are muted.
Thank you, Chiara.
Okay.
So 15 years flew by really fast. And I remember exactly when Nature Nanotech actually got on my radar. You know, I'd started my lab back in India, and it was when I picked up what I think was your third issue. I saw this paper by Thomas Carell, and I thought, yep, this is the kind of work I want to do. And from then on, I always found myself waiting for the next issue to see more such work that was always either cool or neat or exciting. And so to the Nature Nanotech team, I just want to say congratulations on doing that for every issue for the past 15 years. And so I picked a quote for you today to celebrate this early landmark, right? And it goes, excellence is never an accident. It's always the result of high intention, sincere effort, and intelligent execution. And it represents the wise choice of many alternatives. And here's looking ahead. Choice, not chance, determines your destiny. And if I sound like Aristotle, it's because I'm quoting him. Okay, so for today, I was asked to talk about DNA nanotechnology and its potential applications in medicine. And because of the fact that the field is actually so broad, I'm going to restrict my talk to the kind of DNA nanotechnology that deals with precision architectures, where you know the number of strands and you can count the number of strands in a given architecture. So this field, called structural DNA nanotechnology, started really with Ned Seaman constructing a DNA cube from 10 strands of DNA. And when matching strands paired with each other, the resulting helices stiffened up into rods, forming a molecule that would have had the shape of a cube. But this was done in only about 1 to 3% yield. And so the next leap came when Andrew Turberfield at Oxford made a tetrahedron, but with about 98% yield. Now we were approaching systems that would enable practical applications, since they could be made efficiently. Then the next step change, in my opinion, came when Paul Rosamond used a method called DNA origami to fold an extremely long strand of DNA into virtually any desired shape in 2D. And this method works by choosing a set of smaller strands called staple strands. And when you add these staples into a solution containing the longer strand, they latch onto specific locations of the longer strand and pinch them together in space. And really where you make your connections decides what shape you can fold this longer strand into. And one can now do this not only in 2D, but you can also do this in 3D. Suffice it to say, you can now basically do 3D printing on the nanoscale using DNA as a material. So in parallel, a different class of precision architectures based on DNA was emerging. And here, small modules of DNA, be they rigid rods or triangles like so, could be made to undergo specific movements, switching from one conformation to another. And here the movement is powered by the addition of a stimulus that could be DNA strands or small molecules or chemicals that upon binding the architecture, either pinched it or relaxed it, or changed the twist of the DNA helix. One could even use the space enclosed by DNA helices to encapsulate cargo and then release it by changing the dimensions of the DNA helix. By sort of changing the dimensions of the surrounding polyhedron, adding specific in response to some specific strands. So you can see now that complex DNA systems can be used to template other functional materials and thereby leverage their signature capabilities. So in general, if a material can be chemically conjugated to DNA, then a DNA structure can both inherit the high performance properties of that material, but it can also offer a nanoscale level of organization. And this enhances the material's performance, or can sometimes even create an entirely new property like emergent properties. So over the last 20 years, this extraordinary power of DNA to organize functional materials has really become quite apparent. And DNA nanotech already deeply draws on principles and approaches from diverse areas, ranging from traditional material science, physics, chemistry, biology, computation. But what I find most exciting is that DNA is actually an ideal scaffold at the biotic-abiotic interface. Not only is it water soluble, it can recognize and even communicate with biological molecules, and it's highly engineerable. So, although DNA nanotechnology encompasses many areas of interest, including material science and computation, I will discuss today how we can interface DNA nanodevices with living systems and try to join the dots from discovery biology to precision medicine. Okay, now this is how I have found most chemists, and in fact, even most biologists view cells, right? You have the cell membrane, you have the nucleus, you have the cytoplasm. Naturally, many of the things they wish to target also reside at these locations, right? So on the cell membrane, you have ion channels and GPCRs. In the nucleus, you have DNA that they wish to target. In the cytosol, you have RNA and proteins that they wish to target. And yes, chemists and biologists also recognize that different cell types exist. And indeed, this has led to them recognizing that disease and dysregulation can be pinpointed now to specific cell types as part of organs that malfunction, right? And this naturally led Bob Langer, including many others, to delineate the concept of drug targeting, that is, enriching a drug at a tissue of interest. And then this leads to localized corrective action at the tissue level. In fact, we had a wonderful inaugural lecture by Bob Langer, who outlined various ways by which one can achieve targeted delivery at the tissue level, where triggered or controlled release achieved remarkable therapeutic efficacy. And we now know that all of these processes are regulated by organelles within the cells of the affected tissue, and that dysfunction of the tissue can be compensated for by toggling the functionality of organelles in cells that are defective. And so I posit that there will be much to be gained from achieving an additional level of drug targeting, where we not only send drugs to specific cell types, but to also specific organelles within those target cells. And so I'm going to suggest that we all start viewing cells the way cell biologists always have, with all of their organelles, if we are to solve the next set of currently unsolvable problems. And here's one such currently intractable problem that I think DNA nanotech is likely to deliver a solution for. We're not there yet, but we seem to be getting there. We didn't think our DNA nanodevices had such an application when we started out, but I'll try to connect how we started out and how we got to this point. Again, it's really nice to have Hagan end his talk saying that it was basic science that really propelled these very new applications in sequencing. And that's likely to be the case even in this particular thing in the future. So first, we wanted to see if we could localize DNA nanodevices, not just tissue specifically, but target them to specific organelles within those cells. And I'm going to describe our recent work showing that it is indeed possible to take a DNA nanodevice, reprogram it, and then send it precisely to an organelle called lysosome in 3 different cell types in worms. These are coelomocytes, which are the putative immune cells of the worm, the intestinal epithelium of the worm, and in specific neurons in worms. But first, we wanted to see if it would be even possible to send DNA nanodevices to neurons, which hasn't been done. So for this, we fused a DNA binding protein that is based on a recombinant antibody that we call 9E for some reason. And we fused it to a cell surface receptor, which is an odorant receptor called ODR2, that is expressed nicely on the plasma membrane of neurons. So if you inject DNA nanodevices into the pseudocoelom of the worm that expresses this fusion protein only in its neurons, then the nanodevices exclusively label the neuronal membrane, plasma membrane surface. And so clearly now we have cellular level specificity, but can we achieve organelle level specificity over and above this? So if you fuse 9E to a protein like synaptobrevin, which is 5% on the plasma membrane and largely present in endolysosomes, and you tissue specifically express this fusion protein in specific neurons like either cholinergic neurons or the glutamatergic neurons. And if you inject nanodevices now into the pseudocoelom, they will specifically label endolysosomes only in whichever set of neurons is expressing this synaptobrevin 9E fusion protein. And so far, I've shown you that we're using synthetic biological parts, right? The DNA binding protein 9E is based on a recombinant llama antibody. Therefore, you need to use transgenic worms. So I will show you now that you can achieve tissue-specific and even organelle-specific labeling, even without genetic modification in wild-type worms, provided you have a natural receptor that you can tack onto. For example, if you look at intestinal epithelial cells, they express a double-stranded RNA binding protein, which acts like a receptor for double-stranded RNA, called SID2. In fact, it's thanks to this receptor that RNAi works so well in worms. So now if you attach a ligand for SID2, which can be a simple double-stranded DNA module that's about 50 to 100 base pairs long, if you attach it to a DNA nanodevice and then introduce the DNA nanodevices into the worm gut, these nanodevices now nicely label the lysosomal organelles in specifically your gut epithelium. So I spent a bit of time trying to look for molecular platforms and scaffolds which you can introduce artificially, extraneously, into a multicellular organism. And these artificial scaffolds can then achieve both tissue level specificity as well as organelle level precision. I found very few that do so with tissue level precision that I include over here. Many of them are familiar to us. But I could only find GFP technology as well as natural pathogens or synthetic virus-based technologies that achieve both cellular level precision as well as subcellular level precision. So in case I've missed out others that you can think of, please let me know. I would be very grateful for more examples. I've been trying to look for them. Okay, so now DNA nanodevices joins a very small number of high precision technologies for biological targeting. And I believe these high precision tools for biological manipulation will enable us to uncover exciting new basic biology, as well as prove useful for the next generation of organelle-targeted therapeutics. And so here's what our lab has been trying to achieve by trying to interface DNA nanodevices directly with living systems, right? So we already know that cell state is connected to cell function. Let's take, for example, an immune cell in its resting state. If it's starving, it upregulates a process called autophagy, and it activates a specific transcription program that tips the cell into a state that is different from the resting state. If it encounters a pathogen, this resting cell will suddenly activate inflammation, and this sets off a different set of transcription programs for immune activation. Now, we already know that organelles in the cell rise to meet each of these distinct challenges. They change their function to help drive the cell's overall new function, right? So during autophagy or inflammation, an organelle called the lysosome in immune cells senses either starvation or it senses the pathogen and sends an appropriate signal to the nucleus, which then initiates the signaling. And importantly, lysosome biologists like David Sabatini, Sergio Grinstein, and Juan Bonifacino have shown that the nature of the lysosome itself changes in each of these cell states. So what I'm trying to ask here is the reverse. We know that there must be some feedback between the nucleus and the lysosome, right? Because once the pathogen is cleared, or once the nutrients are now available, the lysosome has to communicate this change in state that it's sensing to the nucleus in order to go back to its resting state and change the transcription program again, right? So here's what my lab is trying to see. We're trying to see if we explicitly change organelle state artificially, can we change cell state? But how do we define this organelle state, right? And I'm going to posit that we can map organelle state by measuring the ionic composition of the lumen of the organelle. So I'm going to start by asking 3 questions. 1, can we define organelle state using such an approach? 2, does it change appreciably in health or disease? And finally, can we change cell state if we change the state of the organelle itself artificially? So if you want to measure an ion in an organelle, you need a device that can perform 2 functions. The first function is to measure your ion of interest. So one portion has a measuring module. So the measuring module can either be an ion-sensitive dye, or an aptamer that binds a small molecule, or any kind of chemical sensor that's either a turn-on sensor or a turn-off sensor. Now, because you can append a ratiometric fluorescence dye in a precise 1:1 stoichiometry, because Watson-Crick-Franklin base pairing is 1:1 of 2 strands, this detection chemistry becomes quantitative inside a living system. Okay, to measure your ion inside an organelle, you need to be able to target this measuring device into the lumen of that organelle. So the second function is that the probe needs a targeting module. So for this, we use the principles of intracellular trafficking. First, you identify a shuttle protein on your target organelle, and a shuttle protein has 2 characteristics: a high abundance on your target organelle and a low abundance on the plasma membrane. The second is that there's an active exchange between these 2 populations. So if you stick on a molecular motif onto your nano device that gives it an ability to latch onto the population at the plasma membrane, it can hitch a ride into the organelle. And because of the active cycling of the shuttle, the probe or the nano device gets nicely enriched into your organelle. And by fluorescence imaging, you can simply read out the concentration of your ion. So this entire platform, building it, is still a work in progress, but we've been able to measure a few ions in some organelles, and we've shown them in green here. But we still have some more left to measure and a few more organelles to reach, and these I've shown in maroon here. But with what we have so far, I'm going to ask the first 2 questions of whether we can use ions to read out organelle state and whether these states differ in health and disease, and if they are, whether that difference is even measurable. We're going to try and see if we can measure ions with DNA devices, and for that, I'm going to introduce the first nano device called the iSwitch that was developed by my very first grad student, Shobik Modi, and it acts like a pH sensor. So at acidic pH, it undergoes a shape change, and this can be visualized by sticking a fret pair onto the assembly. Now, this turned out to be a very nice ratiometric sensor if you plot the fluorescence of the donor fluorophore divided by the acceptor fluorophore as a function of pH. The second is a ratiometric chloride sensor that is completely insensitive to pH, and this was made by Sonali Saha. And what she showed was that if you take one DNA strand that's carrying a chloride sensitive dye called BAC, and a complementary strand that carries a normalizing dye like Alexa 647 that is insensitive to chloride, then this entire nano device acts like a ratiometric chloride sensor. So now if you add either of these nano devices to the cell culture milieu, and if your cells are expressing a receptor called the scavenger receptor, this nano device will act like an endocytic tracer. Simply replace the cell culture media with, add the device to the cell culture media, replace the cell culture media with fresh media, wait for 5 minutes. Your endocytose device will then label the early endosome in 5 minutes. If you wait an hour, it labels the late endosome. And after 2 hours, it's in the lysosome. So now, after you label your endosome stage, whichever specific endosome you want to with the probe, you can fluorescently image your biological sample in 2 channels, right? And so if you use a pH sensor like the iSwitch, you can get a pH heat map. And this shows you the pH in literally every endosome you labeled. If you use a chloride sensor instead, in a similar way, you can get a chloride heat map. Interestingly, although people had measured pH inside lysosomes, nobody had been able to measure chloride inside lysosomes because these organelles are so acidic that all conventional methods to fluorescently image chemicals in lysosomes actually fails. And in fact, the first ion actually that was measured inside lysosomes was pH. And this was done, I think, 135 years ago by Eli Mechnikov. And the second ion that was measured inside the lysosome was chloride. And that was achieved by actually a DNA nano device, and in 2015, after a very long time. So at this point, we just didn't ever imagine that DNA nano devices would turn out to be capable of, subtyping a neurodegenerative disease or assessing the cellular responsivity of a patient's cells to a drug. But I want to tell you how we ended up there from this point. Okay, so far I discussed taking a single sensor, measuring a single ion, and getting the distribution levels of a single ion in a collection of lysosomes. Then we thought, if we take a combination sensor that can measure 2 different ions in the same lysosome, such that we could tell the concentration of, say, pH and chloride for every single lysosome, would we be able to see if we could resolve these populations inside a cell into different populations? And the reason why we thought this would be cool was that's how 2D gel electrophoresis or 2D NMR works, where species that are hard to resolve in one dimension end up resolving along a second dimension if the second dimension is something that is a completely independent property from the first one. Okay, so Kaho, a postdoc in our lab, made this combination sensor by sticking a chloride sensing dye onto our pH sensor. So now you have 3 strands, each carrying a dye. We'll be imaging 3 dyes now, and the ratio of the green to the red dye gives you chloride, and the ratio of the orange to the red dye gives you pH. And so here's what typical data will look like. Here is a cell that's been labeled with this combination sensor, but we've artificially fixed the chloride levels of these endosomes to 5 millimolar and the pH to 4. So we expect all the endosomes values to cluster around a specific point on a 2D plot. that's exactly what happens, but there are a huge number of points lying one on top of the other here. So this is not a great way to visualize this. But if you convert it into a density plot like shown here, then you see a very nice focused point, and that shows that this method works quite well. And we call this imaging method 2IM, which is short for 2-ion measurement, and a plot like this is called a 2IM profile. Okay, so if you look at the 2IM profiles of lysosomes in skin biopsies or skin fibroblasts that you can get from a skin biopsy from a healthy individual, you see that the lysosomes can be resolved into two distinct populations. And here's showing the data for 3 different healthy people, and they all show this high chloride, high acidity population here. And this changes dramatically when we do a 2IM profile in skin biopsies of patients with Niemann-Pick disease. Now, Niemann-Pick disease comes in at least 3 flavors called Niemann-Pick A, Niemann-Pick B, and Niemann-Pick C. In Niemann-Pick A and B, the same enzyme, acid sphingomyelinase, is mutated. A is a much more severe phenotype than B. Niemann-Pick C, on the other hand, has a completely different protein that's mutated. And in all these diseases, you see that this high chloride population is completely lost. And so what we then did was add the therapeutic for Niemann-Pick A and B. They share the same therapeutic, which is a recombinant enzyme of acid sphingomyelinase. And when it's added to cells, this enzyme is endocytosed, it traffics to the lysosome, and it sets right the milieu in the lysosome. And so when you treat cells derived from patients with Niemann-Pick A and B with acid sphingomyelinase, you see that this high chloride, high acidity population now suddenly re-emerges. For the Niemann-Pick C population, there's actually no therapeutic yet for these people. There is a molecule called beta-cyclodextrin, and everyone knows it's not a fantastic therapeutic, but they are still taking it through clinical trials because these poor people don't have anything else. And so when you treat Niemann-Pick C cells derived from 3 different patients with beta-cyclodextrin, you can see this guy is a responder, this guy is sort of borderline, but this guy is a non-responder. Note that if you take Niemann-Pick A cells and treat them with beta-cyclodextrin, this is the wrong therapeutic for these people, okay? There is no response at all. And so the NIH just started a clinical trial for Niemann-Pick C disease, and they wanted to use this test to see if we could get either positive predictive values or negative predictive values for patient response. So I'm quite excited by what we're going to see. So clearly now we have a way to assess if a person who's suffering from neurodegenerative disease, if their cells will respond to a therapeutic or not. And we can potentially use this 2IM method to pre-assess patient suitability or distill out suitable patient cohorts for clinical trials for neurodegenerative diseases. And we know that these drugs really have trouble clearing, meeting these efficacy because there's sometimes no rhyme or reason why people respond and why they don't. Finally, I'd like to address the third question of whether we can change cell function by changing organelle state. We recently serendipitously discovered that a DNA nanodevice that displayed an aptamer for the Ku protein, when it was added to immune cells called macrophages, ended up kickstarting a signaling pathway that causes very nice, normally spherical lysosomes inside macrophages to suddenly elongate into these big tubular forms called tubular lysosomes. So we use DNA nanodevices for pH and calcium and enzyme activity to understand what is it that is different between these classical vesicular lysosomes that we see here and these tubulated lysosomes. And what we found was that all along the length of these tubular lysosomes, there was a gradient of both pH as well as calcium. In fact, Proteolytic activity, the ability for lysosomes to hydrolyze and degrade materials, was much higher in the vesicular form and much lower in these tubulated forms. We also found that these tubular lysosomes were responsible for secreting more amount of an enzyme called MMP9. And it was MMP9 that was, by a feedback mechanism, initiating this particular signaling cascade and causing the lysosomes to tubulate even more via a positive feedback. And these tubules were essentially pushing the cell membrane while the MMP9 enzyme was causing extensive membrane ruffling. And together, they made these macrophages much more phagocytic than they started out with. So actually, changing the organelle state explicitly can change organelle function. As you can see here, we go from vesicular lysosomes to tubular lysosomes. And by changing organelle function, we can change cell function. We leverage this capacity of DNA nanodevices to enter organelles specifically in specific cells and change their state, and thereby change cell function, even for therapeutic effect. And a paper regarding this is going to be online tomorrow at Nature Nanotech in the ASAP column. And critical to that application, you will see online tomorrow, was the ability to target DNA nanodevices in a live organism to specific cell types. But over and above that, we achieve subcellular level precision, targeting it to specific organelles, which we had already developed in live nematodes. I've been very lucky to have worked with some incredible graduate students, postdocs, and really wonderful collaborators on a rather unconventional direction for our field. And hopefully we've been able to convince you that if even simple DNA duplexes that have been programmed with a few small molecules can reveal something useful about biological systems, then the complex architectures that the rest of the field is engaged in creating will certainly be capable of so much more. So finally, happy anniversary once again to Nature Nanotech. And I'm delighted for the opportunity to share in this excitement, and I look forward to any questions that the audience may have.
Thank you very much, Yamuna. This was really a fascinating talk and really great. I have one very general question, actually.
Yes.
So the idea is to, apply these DNA probes and DNA nanotechnology to in vivo. So what can you say about the stability of your probes? Is that a challenge? Did you have to specifically devise the probes so that they would be more stable? And will they be applicable to all different type of disease, or in certain diseases where the environment is going to be like different? Do you think that you will need to change something?
Yes, so these are fantastic questions. I think there were two questions loaded into one, but so let me address the question of stability first, right? So it's quite interesting that when we first started taking DNA devices and putting them on living systems, it was clearly a very different thing from what the rest of the DNA nanotech field was doing, and I think they were all a little flummoxed as to what we were trying. And at the time, we just wanted to explore and see what would be possible. The reason why people were always ginger about putting DNA nanodevices or small DNA duplexes onto cells was the idea that biologists have that all DNA is immunogenic, right? And so there's no point in, and they certainly turn out to be immunogenic because there are mechanisms in the cell that snip up DNA. And this is, once it's degraded into these nucleotides, that is the source of the immune response, right? And it also binds TLRs and things like this. But you and I have floating DNA duplexes in our bodies all the time, and we're not breaking out into rashes and having lupus and things like this. And the reason is there is a difference between self DNA and non-self DNA. Our devices are designed to mimic self DNA. And self DNA has a very specific entry pathway in cells, and they have a specific lifetime as well. They do not trigger TLRs. They do not trigger the immune response because the system doesn't want to generate an immune response to its own self. So now, we've shown in a paper in Angewandte Chemie, and actually for every one of our devices that comes out, we always look at the stability of the DNA nano device in that particular cell type. And this is an essential starting process when one has to do carry out work in any living system. We have specific assays that we've published in materials and in sort of methods in molecular biology of how to do this. And it turns out that you actually have a window of time within which you can make your measurements. I also want to contrast this to fluorescent protein technologies. I'm really glad that people are asking us to live up to a very high standard, which is tell us that your DNA devices are, how stable they are they, when do they get degraded, etc. And we've done all of this. But fluorescent proteins are capable of degradation. So when you take CFP, YFP fluorescent proteins, they can also be degraded, and the CFP and YFP can still give you very interesting fluorescent signals that might have nothing to do with your phenomenon of interest. So I always say that please ask these same questions to fluorescent protein reporter people as well, because this is probe degradation inside a system is certainly important, and we have to have ways to measure this. The second question, if I'm not mistaken, was what are the kinds of applications that might be possible? I think anywhere, so most cells that actually take up DNA devices turn out to be, naturally, turn out to be immune cells, right? As well as skin fibroblasts, any cell that has scavenger receptors, because this is a way by which the system's always interacting with dead and floating DNA in the body. Into all of these, you can actually send your DNA devices without too much of a problem. And I think the capacity to achieve immunomodulation with DNA represents a really great outlook. And I think connections between the neuroimmune pathway as well as cancer and immunotherapy, I think these are all, this itself is a very big sort of playground to really show the power of DNA nano devices, because we've shown in a paper in PNAS recently last year that they're exquisitely tunable. The immunogenicity is exquisitely tunable. You can make DNA which is naturally non-immunogenic, but you can make one or two changes on your scaffold and introduce titrated immunogenicity. And so this may be one way where you want to stimulate the immune system or change the immune system's capacity to do specific functions, which could have implications in autoimmune disease and cancer and neurodegeneration.
That's great. Actually, there is a question from the audience that ties into this. So do you see an opportunity for organelle targeted DNA devices for trained immunity induction, given the effects you see in macrophages? For example, would you see an application for developing vaccine technologies?
Oh, I hadn't thought about vaccine technologies, but I think there would definitely be opportunities to be able to train immune cells. We can already show that macrophages are capable of changing their function, right? They become very phagocytic. You can increase degradation in the in a lysosome or not. You can depress degradation in lysosomes, as we showed with this Tudor. So I would not be surprised at all if we could actually engage adaptive immunity soon.
Okay. And another question from the audience is, can your DNA-based nanosystems be decorated with self-specific peptide ligands?
Absolutely. I think this is one of the it's a really terrific application to be able to do that, right? Because not just specific peptides, you can think of specific immunogenic tags that you want to put in specific ratios. Start asking which TLRs are talking to each other, which TLRs are synergistic, which TLRs are antagonistic. So any immunogen or anything that suppresses or can be inhibitory to a specific TLR, all of these applications are possible because you can connect nearly any molecule if you try hard enough to DNA.
That's great. So I think I have another one. Yes, so another question. Sorry, I just need to scroll down and read it. Okay. So someone from the audience is pointing out that DNA-based systems have issues to enter the cell due to the negative charge. And again, they could be degradable by enzymes such as DNases. How can you design the DNA-based sensor or any other kinds of DNA-based biomedical system to overcome these issues?
This is a great question, right? So DNA is actually a substrate for degradation by DNases. You will have a specific lifetime, circulation lifetime, etc. But you know, we have many kinds of ways to bypass natural DNA degradation, right? So one can use LDNA if you want, which is the mirror image of DNA. It would still bind many of these scavenger receptors. John Chaput has come up with triose nucleic acids, which are also negatively charged, also capable of forming DNA helices. And so we can have many different kinds of nucleic acid analogs that are completely resistant to DNases and hang around for a longer time if you want, but still be capable of binding these receptors and being uptaken. My estimate would be that, you know, I generally try to keep things as biocompatible as possible, and unless I need an application that requires this to hang around for a longer time, we generally don't go for these nucleic acid analogs, though we could convince reviewers very easily by, you know, making triose nucleic acid TNAs or any one of these resistant nucleases and then show applicability. But I think there's value in having things not hang around in a system. If it's metabolized harmlessly, then it really serves as a good reporter because it's non-perturbative. So yes, that is a great question.
And so you showed that you've mainly talked about the applications to map lysosomes, chemical state.
Yes.
So what about the other organelles? What are the challenges for those, and what do you think will be the merits in targeting those as well?
Yes, I have a group of only 10 people. So, but I think it's all possible. So you will see a few things coming out on the recycling endosome fairly soon, as well as the Golgi. So these things will take a little bit of time, but applications will be forthcoming. It's just that the lysosome is the easiest organelle to look at. And that's why we started with the lysosome, but we can easily send DNA devices to other organelles. So with Rajni Rao at Johns Hopkins, we're looking at the recycling endosome, and with Brian Delisle at University of Kentucky, we're looking at the Trans-Golgi network. And I'm not going to I'll stop there and just say that applications are coming.
Fantastic. And another question. So the idea would be to move these to a clinical application, and to move this for diagnostics and precision medicine.
Yeah.
So and a lot of issues when talking about this, especially in nanomedicine, is scaling up like making the product so that it's compatible for in vivo and in human application. So what are the challenges for DNA for your DNA probes in this context?
So I think immunogenicity is maybe not going to be too much of a problem, because we're seeing that these are actually non-immunogenic from mice to human, human cells, as well as in worms and zebrafish. I do think that our problem would probably be to make it more stable and increase circulation lifetimes. But again, what we will see is hard. I mean, where things might fail is hard to predict until we actually go and do it. The important thing, the thing that gives me a lot of hope is that there was a time when people thought that a nucleic acid cannot be a therapeutic. But I think Moderna and Pfizer have shown that you can. It is actually possible to treat the globe with nucleic acid-based therapeutic with these mRNA vaccines. So I think that problem has been solved, and we will start seeing a plethora of nucleic acid-based therapeutics where the nucleic acid itself is the therapeutic.
Yeah. Thank you so much. I think we don't have any more questions from the audience unless I missed anything. I don't think so. So really, thank you so much for showing us your work. It's always, I find it always fascinating and really, really interesting. So it was great that you agreed to participate to this webinar series. Thank you very much to the audience as well for listening. And again, you can register for all the other webinars by going also on our webpage, but you'll receive an email after this webinar with the link. Thank you again, Yamuna, and thank you everyone, and
Thank
hopefully
you,
see you.
Cara.
Bye.
Thank you, everyone.